Hello and welcome to my presentation about human immunity. Thank you for coming and I hope you do enjoy. Firstly, I want to talk about the learning objectives. I have sectioned my presentation into 3 different sections. So here we have section 1, section 2 and section 3. I will firstly begin with section 1. So firstly, I want to begin talking about the different types of microorganisms. Microorganisms are also known as pathogens, and they can be the cause of illnesses. And here we have 4 different microorganisms up on the screen. So firstly, I will begin with viruses. Viruses are the smallest microorganism. They can only survive when attached to living humans or animals. They can be spread through indirect contact such as coughing or sneezing. An example of a virus is influenza. Secondly, bacteria are single-celled organisms which can be found anywhere on the earth and even in the lining of the human digestive system. As beneficial as they can be, they can also be quite harmful. Bacteria forms E. coli, which is food poisoning from out-of-date foods. This can make humans extremely unwell. Then moving on to fungi. Fungi can be a single-celled or complex multicellular organism. Fungal infections can be also known as mycosis, and at times can be harmless, but is also capable of causing diseases. Fungal infections mostly affect the skin. They affect the skin by releasing spores which can be picked up by either direct contact or through inhaling the disease. Some examples of fungi infections can be athlete's foot, ringworm, or yeast infection. And lastly, parasites are only able to survive on a host. This enables them to grow and multiply, making it easier to spread diseases. Parasites can spread through food, direct contact, and bloodborne infections. This can be done through insect bites. An example of a parasite is the malaria infection. Now I will move on to the first and second line of defences. I will firstly begin with the first line of defence. This acts as the physical and chemical barrier to help protect the body internally and externally from pathogens. The main source of protection is the integumentary system, as it is the largest organ of the body and consists of the epidermis, dermis, and subcutis. The epidermis acts as a protective layer. It is waterproof, which helps keep bacteria and germs from entering the body, and this aids cell cyclical molecules to help fight harmful microbes away. It also helps heal abrasions and cuts by sending white blood cells to the area. The dermis is important as it consists of hair cells, which protect the body from harmful pathogens by blocking the entry. So the hair on our nostrils helps protect the respiratory system as it keeps out dust and other particles harmful to the lungs. Now, when we are speaking about chemical barriers, this can consist of the mucous membranes. They are coated with secretions that help fight microorganisms. For example, lysozyme is found in tears, sweat, and saliva, and this destroys pathogens when they are trying to enter the body. They are also very good at providing nutrients. Here on a diagram, we can see different examples of chemical barriers. So here we have the vaginal acids in the vagina, and then we also have pH changes and also mucus linings in the trachea. However, if pathogens do successfully pass the first line of defense, the second line of defense begins. This is also known as the innate system. The innate defenses are non-specific. This means that the body responds in the correct manner and the same cells and proteins no matter what pathogen. There are also cells called pathocytes, which we can see on the slide that I have up now. These live in the bloodstream, and some examples of pathocytes are the macrophages and neutrophils. When pathogens enter the body, the pathocytes are alerted of this, and phagocytis then kicks in to destroy the pathogen presence. It begins with the macrophages identifying and binding themselves to the pathogen to engulf and break it down with the help of lysosomes to destroy it. All while this is happening, the macrophages are alerting other proteins called cytokines. This brings in more leukocytes such as neutrophils, endocytes, and basophils, as we can see on the slide here. Neutrophils make up to about 40 to 70% of the white blood cells and help with the phagocytosis process, and the endophils make up about 5%. These contain toxins which can kill pathogens if they are too large to be engulfed. They also release proteins to help support the inflammatory process. And now here we do have on the slide a diagram of the inflammatory process. It firstly begins with the release of histamine when injured, and then phagocytes migrate to the area, which then will be able to engulf any diseases and pathogens present. Once this has happened, phagocytes will move out of the area to allow the capillaries to disperse platelets to be able to clot the blood and begin healing the area that was injured. I will now be moving on with section 2. So here we have the third line of defense. The third line of defense contains cells that destroy specific pathogens. These cells are called dendritic cells, and they are a communicator between the innate and adaptive immunity systems. The adaptive immunity response takes place in the lymph nodes and involves B cells and T cells. These can be found in white blood cells, and this is able to mature the antibodies. The process is when dendritic cells are in the lymph nodes, they are searching for the T cells. The T cells then can help then destroy the pathogen present. Once this has taken place, memory T cells will remember this specific pathogen and respond immediately if it ever does invade again. I will now be moving on to the different types of immune cells. So here we have a picture that is showing you what these white blood cells look like. And we also have a table on the left side which is giving you the different names of the white blood cells. So here we have neutrophils, and their function is they are the most innate due to being able to phagocyte pathogens. Then moving on to the endophils and the basophils. These are important to defend against parasites and involved with allergic reactions. Monocytes help to develop into macrophages, which then enables them to engulf and destroy pathogens. Lymphocytes, which these link into the B cells and T cells, which I will go on to explain. These assist with fighting infections and disease, and they do contain of the B cells and T cells. These are mostly found in the bone marrow. So B cells are made up of antibodies which help fight pathogens, while T cells help to fight and kill cancer cells and controls the immune response by killing infective cells. Moving on to immunoglobulins. There are 5 different types of immunoglobulins. This is the IgG, IgM, IgA, IgE, and IgD. Immunoglobulins is made up of 4 chains, and 2 of these chains have a higher mass which allows the immunoglobulin molecule to spread into the 2 heavier chains and lighter chains. And they are always normally arranged into a Y shape, which we can see on this slide. Firstly, beginning with the IgG. IgG is most common and is present in large amounts of blood and tissue. They are easily transferable during pregnancy. This could be through breast milk, and they can attack most of all pathogens. IgM is commonly present as an antigen. It is made up of B cells and is effective at attaching antigenetic determinants of the outer coats of bacteria. IgA is mostly found in tears, sweat, and bodily secretions. This means it is located in the mucous membranes, and it is produced by B cells, but not as stable as IgG cells, meaning that it is present in lower amounts. IgE is made up in small amounts of B cells within the blood, but when pathogens or allergies are present, there is an increased response to help protect the immune system. And lastly, IgD is quite common and acts as a receptor for B cells, and it is only released into the body in small amounts. There is an unclear idea to the functions of IgD. So far, we know how the immune system attacks pathogens which enter the body, but we haven't explored how the immune system doesn't attack our bodies, especially self-antigens. The immune system usually doesn't attack our body cells, tissues, or organs. When the body is faced with antigens which it hasn't seen before, it will be recognized by the adaptive and innate systems, and they will be able to respond accordingly. And there is also other two important factors which allow the immune system to tell apart of the self and non-self, and these are the major histocompatibility complexes, IHC1, MCH1, and MHC2, alongside the human nuclear antigen, HLA, gene complex as well. For example, the transplantation of a donor organ must have the major histocompatibility complexes or the human-like leukocyte antigen gene complex matching, and this will be able to reduce the chances of rejection. If there was a failure of self-tolerance, it can be described as an autoimmune disease. There is not a specific reason to why this occurs, but it can be due to a combination of genes and damaged tissues causing the release of antigens. There is 4 different types of self-tolerance and hypersensitivity. The first one is the immediate hypersensitivity, which happens immediately and is produced by an IgE antibodies, and this is known as anaphylaxis or an allergic reaction. Antibody-mediated hypersensitivity, type 2. This is when IgG and IgM antibodies are released and directed to antigens. This can be the cause of phagocytosis and inflammation. Immune complex mediated hypersensitivity, type 3. This is like type 2, but there is a release of antibodies and antigens in the vascular walls and tissues causing an inflammatory response. This can be seen in arthritis. Cell-mediated hypersensitivity, type 4. This occurs when there is an excessive amount of immune reaction of T cells, and this can be linked to Crohn's disease. And then lastly, I have here the rejection of organ transplants. So firstly, beginning with what an organ transplant is. Organ transplant is when cells, tissues, and organs are removed from one individual and transferred to another individual to prevent transplant rejection. Doctors must ensure that the organ is the same match of individuals, but this can be difficult at times. Even if the match is similar, there will be a chance that something is not too similar. But there is something called the monoclonal antibodies which can be used to prevent rejection by removing these T cells. There are 3 different kinds of rejection. There is the hyperacute, acute, and chronic. Hyperacute almost happens immediately. Acute can happen over a couple of weeks or months, and chronic can happen from months to a couple of years. I will now be talking about autoimmune disorders, and this happens when the body's immune system is under attack and destroys all healthy tissues. The first non-specific immune system disorders I will be talking about is rheumatoid arthritis. A rheumatoid arthritis is one example of an autoimmune disorder. This disease can happen at any age due to the body mistaking the healthy tissues as harmful, leading to its attack. Infection, genes, hormone changes, and smoking have all been linked with this disease. This disease affects joints in the hand, leading to stiffness, pain, and fatigue, as seen in this picture on the slide. It can cause deformity of joints in the hand, making it difficult to use the hand and function. To diagnose this disease, doctors will complete a blood count and even X-ray the joints, and there is some X-ray the joints. There is also some treatments for a rheumatoid arthritis. This includes medicine, physiotherapy, or even surgical requirement if needed. The second type of a non-specific immune system disorder is multiple sclerosis. This affects the central nervous system and the myelin sheath. It is damaged by inflammation and scarring from antibodies, viruses, gene deficits, and environmental factors all can play a role into why MS occurs. There is also no true diagnosis as to why it does happen. Individuals are more likely to develop multiple sclerosis if there is a family history of MS. As the antibodies are attacking the central nervous system, the symptoms can vary depending on the location it is being attacked. The symptoms can take place throughout the body, and there can be a loss of balance, muscle spasms, reduced movement, bladder and bowel incontinence, sight, hearing problems, and slurred speech. To be able to diagnose MS, their doctors will conduct MRIs on the central nervous system and even perform a nerve functional study. Treatment can only slow down this process, but there can be medicines such as steroids to be able to help that, and also therapies such as physiotherapy, occupational therapy, and speech and language therapy to be able to help people who suffer from multiple sclerosis. I will now be moving on to section 3. So here we are talking about the acquired immunity. This could actually link into the third line of disease, as acquired immunity is when immunity is developed over time, and this can be through exposure, vaccination, or antibodies. And this can be split up into 4 different categories. So beginning with the natural immunity. So this develops through exposure of pathogens. The body will respond with T cells latching onto the pathogen and present it to the B cells for it to be able to attack the pathogen present. This will allow B cells to remember that specific pathogen and to be able to destroy it the next time it is present. Sometimes these infections or diseases can be passed down from the mother during birth. Artificial immunity is given to individuals purposely through vaccination or immunoglobulin treatments. The same process will occur in natural immunity. The only difference is the exposure is intentional. Passive immunity is only developed once antibodies have been received from someone or something that is only short, and this is only short-lasting and doesn't allow the body to respond the same next time the same pathogen is present. There are 2 different types of passive immunity: maternal antibodies, which can be transferred from mother to child, and immunoglobulin treatments. Antibodies are man-made in a lab or they are extracted from other individuals. Active immunity is the most common type of immunity. A response is developed when an infection or a disease is present. T and B cells are then able to attack this pathogen, and this activates the immune system to enjoy the pathogen present. So when the specific pathogen is present again, the immune system will be able to respond immediately. So why does the immune system respond to vaccinations? Well, the reasons why vaccines are given is so when the immune system has pathogens present, it alerts the white blood cells to begin the primary response. Macrophages and B cells will identify the pathogen, causing plasma cells to create antibodies which will destroy the pathogen. Throughout this process, memory cells have been created. The memory cells are important. If there was ever a secondary attack, the macrophages and B cells will have remembered the previous attack and perform an immediate attack again, and this is called immunological memory, as seen on the slide. Vaccinations can be made up of live annotated vaccines, long-life vaccines, toxoid vaccines, and dead vaccines. So now moving on to the concept of herd immunity. So the reason of why herd immunity is so important is that it's a way to introduce protection from contagious diseases and viruses. When herd immunity is achieved, it protects non-vaccinated and vulnerable individuals such as newborn, pregnant women, and cancer patients. This can reduce the chance of diseases and viruses spreading. For herd immunity to be effective, 70 to 90% of the population does need to be immune. However, there can be some disadvantages to herd immunity. As some of these vaccines do require to have a second booster, this can demotivate people in getting their second vaccine, just due to the side effects that the vaccines can cause. And when people don't get their booster, this can make immunity less effective, meaning that the spread of diseases will be amplified. Lastly, I want to discuss the benefits of vaccinations. So the benefits of vaccinations is that it aids protections to us. When vaccinated, it can prevent up to 3 million deaths worldwide every year. Ever since vaccinations have been introduced, they have prevented death and disabled illnesses such as smallpox. This illness was eradicated by 1980. Another benefit to vaccination is that it relieves pressure from the NHS. As individuals who are not needing treatment for antibiotics, it can be costly. This can also reduce the amount of antibiotics which are being developed and given out. Thank you ever so much for listening to my presentation. I hope you do enjoy it. Thank you.